Okay, let's unpack something truly fundamental to life itself, the cell membrane. It's, the ultimate boundary, but it's actually far more than just a simple wall. Today, we're diving deep into its secrets, our mission. Well, it's to transform your understanding of these incredible structures. We want to reveal how a seemingly simple lipid film becomes this bustling, dynamic hub of cellular activity. It's packed with surprising mechanisms, things that keep every living cell alive and kicking. And we'll be drawing heavily from the definitive guide, Molecular Biology of the Cell, to give you, well, a shortcut to being really well informed. So at the very heart of every single cell membrane is something called the lipid bilayer. Now that sounds like maybe a basic structure, but it's anything but static. How does it even form in the first place? And what makes it so uniquely suited for life?
What's truly fascinating here, I think, is that the lipid bilayer's fundamental structure arises almost magically, spontaneously. It's solely from the properties of its basic building blocks, the lipid molecules themselves. And these aren't just any molecules, they're amphiphilic. That means they have this kind of split personality. They have a water-loving or hydrophilic head, and then these water-fearing hydrophobic tails.
Okay, amphiphilic, got it.
Yeah, and think about it this way. When you mix oil and water, right, they separate. That's because water molecules, they're very social, they prefer to bond with each other. So if you try to force a hydrophobic molecule, like oil, into water, the water molecules have to awkwardly organize themselves around it. They almost build these ice-like cages. And that forced order, it actually costs the system energy. The lipids, they just want to avoid that cost. So to minimize this energy expense, the hydrophobic tails of these amphiphilic molecules, they cluster together, basically shunning the water. And this intrinsic drive causes phospholipids to spontaneously form structures, maybe little spheres called micelles, or if they're shaped more like cylinders, the double-layered sheets we call bilayers.
So it's not just forming by chance, it's practically forced into existence by physics, just to avoid that messy interaction with water. And the mind-blowing implication of that same drive means the torn membrane doesn't just stay torn, it instantly repairs itself.
Precisely. It's like magic, but pure physics.
Keeping life intact.
Exactly. The moment a tear exposes those water-hating tails to the surrounding water, it's energetically unfavorable, very unstable. So the lipids instantly rearrange, sealing the gap. This means the only stable way for a bilayer to exist is by closing in on itself and forming a sealed compartment. And that's an absolute fundamental requirement for any living cell.
That self-organizing principle is just captivating.
Yeah.
But okay, if it's so fundamental, how does the cell stop it from just forming a big random blob? How does it actually harness this spontaneity for, precise function? And what are the actual building blocks? What are the major lipid types we find in there?
Right. Well, lipid molecules make up about half the mass of most animal cell membranes, give or take. The most abundant are the phospholipids. These have that polar head, including a phosphate group and 2 hydrocarbon tails. These tails are typically fatty acids, usually between, say, 14 and 24 carbons long. And often, one tail has a bend or a kink because of the cis-double bond, while the other tail is straight, saturated. These subtle differences in their tails, the length and the kinks, they directly influence how fluid the membrane is.
Ah, okay, so the shape of the lipids matters.
Absolutely. And in mammalian cells, we mainly find 2 main families of these phospholipids, glycerophospholipids and sphingolipids. You can think of them as slightly different chemical recipes, maybe, but they both achieve the same amazing feat of forming the bilayer. And beyond those, membranes also contain glycolipids. These have sugars attached instead of a phosphate head group. And sterols. Cholesterol is the really important sterol in animal cells.
Cholesterol often gets a bad rap, but it's crucial here.
Totally crucial. It inserts itself into the bilayer, kind of nestling its small hydroxyl group near the phospholipid heads, and it plays this dual role. It stiffens the nearby hydrocarbon chains, which makes the membrane less permeable to small water-soluble molecules. But paradoxically, it also prevents the chains from packing too tightly and becoming rigid, so it actually helps maintain fluidity, especially at lower temperatures.
So the membrane isn't just a static barrier. It's this incredibly dynamic fluid environment. How fluid are we really talking? Is it like water or something else?
It's genuinely a 2-dimensional fluid, and that's crucial for almost all membrane functions. Imagine this. Individual lipid molecules rapidly swap places with their neighbors within their own layer, like 10 million times per second.
10 million.
Per second. It's incredibly fast.
This rapid lateral diffusion means an average lipid molecule can zip across the entire length of a large bacterial cell in just about 1 second. They also spin very quickly around their own axis, and their tails are constantly wiggling and flexing.
Wow.
However, a flip-flop where a lipid actually moves from one side of the bilayer to the other, that's incredibly rare. It typically takes hours because that water-loving head group just does not want to pass through the oily, water-hating core. Cholesterol is a bit of an exception. Its head is smaller.
Okay, so zipping around side to side is easy, but flipping over is hard. That makes sense. This fluidity sounds absolutely critical. Does a cell actively control how fluid its membranes are? Or does it just happen?
Oh, absolutely, it controls it. Membrane fluidity is precisely regulated. It's really a matter of survival for the cell. Think about organisms like bacteria or yeasts. Their temperature might fluctuate wildly depending on the environment. So their cells adjust the fatty acid composition of their membrane lipids to maintain constant fluidity.
How do they do that?
Well, for instance, as temperatures drop, they start synthesizing fatty acids with more of those cis double bonds, more kinks in the tails. Those kinks make it difficult for the lipids to pack together tightly, which prevents the membrane from stiffening up or freezing. And eukaryotic membranes, well, they're even more complex. They can have hundreds, even thousands of different lipid species. All of this variety helps ensure the membrane stays perfectly fluid, just above its solidification point, and that also helps membrane proteins fit in and function better.
That level of fluidity is just astounding. But it makes me wonder, if everything is moving so fast, how does a cell maintain any kind of structure or specialized function within the membrane? Surely can't all be just randomly mixed. Can membranes have, like, specialized areas?
That's an excellent point, and it gets into something really interesting. Well, the forces between individual lipid molecules aren't strong enough to hold them in large, fixed groups on their own. The concept of specialized domains, often called lipid rafts, has been a topic of, well, quite a bit of debate in cell biology for a long time.
Lipid rafts, like little islands?
Sort of. In living cells, we don't typically see large, permanent lipid regions like that. It's more like temporary dynamic clusters, maybe tiny nanoclusters, sometimes larger structures like things called caveolae. These microdomains are often enriched in cholesterol, sphingolipids, and glycolipids. And the idea is they act like staging areas. They help organize and concentrate specific membrane proteins together for functions like transporting molecules or relaying signals more efficiently. Protein interactions play a big role here, too.
Okay, so dynamic organization rather than fixed islands. Makes sense. Now, speaking of specialized lipid arrangements, cells store excess lipids in something called lipid droplets. How are those different structurally from, say, the plasma membrane bilayer?
Ah, that's a great distinction. Lipid droplets, which are super abundant in most eukaryotic cells, especially fat cells, adipocytes, they're designed specifically to store neutral lipids, things like fats, triacylglycerols, and cholesterol esters. Because these storage lipids are exclusively hydrophobic, totally water-hating, they naturally aggregate into three-dimensional spheres or droplets, not flat bilayers.
Right, oil and water again.
Exactly. So to exist happily inside the watery cytoplasm, their surface is uniquely surrounded by a monolayer, just one layer of phospholipids. And these phospholipids cleverly orient themselves with their water-hating tails facing inward towards the oily droplet core and their water-loving heads facing outward towards the watery cytosol.
A monolayer, not a bilayer. Interesting.
Yeah, and these droplets actually bud off from the membrane of the endoplasmic reticulum, which is where the enzymes for lipid synthesis are located.
So we've talked about fluidity, specialized domains.
Yeah.
What about asymmetry? You mentioned the bilayer isn't the same on both sides. Why does that difference between the inner and outer layers matter so much?
Oh, this asymmetry is profoundly important for function. It's not just a minor detail. Take human red blood cells, for example. It's a classic case. Most of the choline-containing phospholipids, like phosphatidylcholine, are found on the outer surface, while those with the primary amino group, like phosphatidylserine, which carries a negative charge, are predominantly on the inner cytosolic side.
So there's an electrical difference, too.
A significant electrical difference across the membrane, yes. And specialized protein pumps, called phospholipid translocators, or flippases, work constantly to maintain this specific distribution. And why does it matter? Well, many proteins inside the cell can only bind to specific lipid head groups found on that inner surface. For instance, protein kinase C, a really crucial signaling enzyme, absolutely requires that negatively charged phosphatidylserine on the inner leaflet to become active.
So the lipids themselves are part of the signaling switch.
Absolutely. And this asymmetry also dictates how cells signal in other ways and even how they manage their own demise. Specific enzymes, lipid kinases, can add phosphate groups to minor phospholipids like phosphatidylinositol. This instantly creates new binding sites on the inner surface that recruit signaling proteins from the cytosol to the membrane. Conversely, other enzymes, phospholipases, can be activated by signals from outside the cell to cleave specific phospholipids, generating short-lived intercellular messengers. And maybe the most dramatic example of asymmetry's importance is during apoptosis-programmed cell death.
Right, cell suicide.
Exactly. Phosphatidylserine, which is normally hidden away on the inner layer, rapidly flips to the outer surface during apoptosis. This acts as a very clear eat-me signal to neighboring immune cells like macrophages, which then quickly recognize and engulf the dying cell, cleaning things up.
Well, a lipid flipping sides becomes a signal for disposal. That's elegant. And we mentioned glycolipids earlier. You said they also show extreme asymmetry.
Yes, glycolipids have the most extreme asymmetry of all. They are exclusively found in the monolayer facing away from the cytosol. So in the plasma membrane, that means they're always on the external cell surface. Inside the cell, they face the lumen, the internal space, of organelles like the Golgi or lysosomes.
Always facing outwards or inwards into organelles.
Correct. Their sugar groups are added in the Golgi apparatus, and they are perfectly positioned on the cell surface to play crucial roles in cell recognition processes. Some complex glycolipids, called gangliosides, even carry a net negative charge. And these external sugar chains, along with sugars on proteins, form this protective layer often called the glycocalyx or cell coat.
Cell coat, right.
Yeah, it shields the cell from mechanical stress and chemical damage. It helps keep other cells at a proper distance, prevents unwanted sticking, and critically, it acts as specific binding sites. These can be for other cells or sometimes, unfortunately for the cell, for pathogens. For instance, the toxin that causes cholera actually exploits a specific ganglioside, GM1, as its receptor to get inside intestinal cells, and that leads to the severe diarrhea associated with the disease.
So the sugars are like the cell's ID tags and also its welcome mat, sometimes for unwanted guests.
That's a good way to put it. They're critical for interaction with the outside world.
Okay, so the lipids provide this amazing dynamic asymmetric stage. But for all the real action, the sensing, the transporting, the intricate signaling, that has to be the proteins, right? They're the workhorses, the performers on this stage.
Exactly. You've got it. While the lipid bilayer forms the basic structural framework in the environment, it's the membrane proteins that perform nearly all of the membrane's specific active tasks. Now, the proportion of protein in a membrane varies wildly depending on what that membrane needs to do. For instance, in the myelin sheath that insulates nerve axons, proteins might be less than 25% of the mass because its main job is insulation. But in membranes that are actively producing ATP, like in mitochondria, proteins can be as high as 75% of the mass.
Huge difference.
Huge. But even then, lipid molecules vastly outnumber protein molecules. We're talking roughly 50 lipid molecules for every single protein molecule in a typical plasma membrane. Lots of lipids.
50 to 1.
Yeah.
Okay. So how do these proteins actually manage to embed themselves in this unique lipid bilayer environment? It seems like it would be, well, tricky.
It is, and they do it in a variety of clever ways, reflecting their incredibly diverse functions. Many are what we call transmembrane proteins. This means they span completely across the bilayer, from one side to the other. Their water-hating hydrophobic regions snuggle up comfortably with the lipid tails in the membrane's core, while their water-loving hydrophilic regions face the watery environments on either side of the membrane, the cytosol inside or the extracellular space outside.
So they're amphiphilic too, in a way.
In a sense, yes. They have distinct regions suited for different environments, and they can cross the membrane in different ways. Some cross just once, often as a single spiral-shaped structure called a no-helix. Others weave back and forth multiple times using multiple inohelices. And some even use a different structure, a rolled-up sheet called a Mohs sheet, to form a channel or pore. We call that a burl.
Alpha helices and beta barrels. Different ways to cross.
Exactly. And what's really neat is that scientists can often predict which parts of a protein are likely to span the membrane just by looking at its amino acid sequence. They use computer programs to generate something called a hydropathy plot. This basically graphs how hydrophobic or hydrophilic different segments of the protein are. A stretch of about 20 to 30 predominantly hydrophobic amino acids is a strong clue that it probably forms an eigenhelix that spans the bilayer.
So you can guess its location from its sequence.
Often, yes. It's a powerful predictive tool. And it's estimated that maybe 30% of all the proteins an organism makes are transmembrane proteins. That just highlights how crucial they are.
30%. That's a lot. Okay, what about proteins that don't go all the way through? Do they just kind of hang out on the surface?
Yeah, many proteins are indeed attached to only one side of the membrane. They don't span it. Some are anchored to the inner cytosolic layer by, say, an amphiphilic eye helix that dips partway into the membrane. Or they might be anchored by one or more lipid chains like fatty acids that are directly covalently attached to the protein itself.
Like a little lipid foot.
Exactly, like a lipid anchor. And what's truly fascinating here is that the attachment of these lipid anchors can be highly regulated. It's not always permanent. This allows crucial signaling proteins you might have heard of, SARSAC kinases or RAS GTPases, to transiently associate with the membrane only when they receive a specific signal. This massively expands the functional repertoire of the membrane.
So they can be called to the membrane when needed. Clever.
Very clever. And then there are other proteins entirely exposed on the external cell surface. They're attached only by a special covalent linkage to a lipid anchor called a GPI anchor, glycosylphosphatidylinositol anchor.
PI anchor.
Okay. It's like the protein has this built-in molecular sticky foot made of lipid and sugar that keeps it firmly anchored to the exterior, but the protein itself never actually pierces the bilayer core.
So many ways to associate with the membrane. You mentioned IBA barrels earlier. How do they compare structurally and functionally to those ivy helical proteins? Are they common?
Abiral proteins are a bit different. They are always arranged as this rigid, hollow cylinder, like a tube or barrel, as the name suggests. They're typically found in the outer membranes of bacteria and also in mitochondria and chloroplasts within eukaryotic cells. Unlike eye helices, which can have some flexibility, the barrels are incredibly stable and rigid. That's because of strong hydrogen bonds linking each strand of the eye sheet together.
More rigid than the helis.
Much more rigid. And many of them function as porins. They form these water-filled channels that allow specific small water-soluble molecules, nutrients, maybe waste products, to cross that outer membrane barrier. And they can be selective too. Their internal loops can actually narrow the channel, acting like a filter, to only let certain molecules pass, like a specialized porin called multiporin that specifically allows the sugar maltose through.
So they're like selective gates.
Precisely. And their rigidity makes them remarkably stable, which, from a research perspective, often makes them easier to purify and crystallize to determine their structure compared to many of their alegal counterparts.
Right. Okay, let's shift back outside the cell for a moment. Cells are constantly interacting with their surroundings. How does that outer surface, the plasma membrane, prepare for all that contact and communication?
Well, as we touched on with the glycolipids, the extracellular face of the plasma membrane, the side facing the outside world, and also the internal faces of organelles, they are heavily decorated with sugars. It's called glycosylation.
Glycosylation. Covered in sugar.
Pretty much. Most transmembrane proteins in animal cells have these short carbohydrate chains, oligosaccharides, covalently bound to them, mainly on their extracellular portions. This creates that cell coat or glycocalyx we mentioned earlier. These sugar chains, along with the sugars on glycolipids and other specialized sugar-protein complexes called proteoglycans, are always positioned outward, forming this kind of fuzzy layer.
Fuzzy layer. I like that visual. What does it do?
It does a lot. It protects the underlying membrane against harsh chemical conditions like low pH or damaging enzymes. It helps keep other cells at a bit of a distance, preventing unwanted clumping. And, crucially, it acts as highly specific recognition sites. This is key for cell adhesion and communication. For example, there are specialized proteins on some cells called lectins, which specifically bind to carbohydrates on other cells. This mediates things like immune cells sticking to blood vessel walls near an infection site so they can get out into the tissue.
So the glycocalyx is protection, spacing, and recognition, all in one.
You got it. It's a really important interface.
Now, studying these embedded proteins sounds like a massive technical challenge. How on earth do scientists even manage to get them out of the membrane to study them without destroying them?
It is incredibly tricky. You're right. You can't just dissolve them in water like you can with soluble proteins because of all those hydrophobic regions designed to sit in the lipid core. So to liberate membrane proteins, to get them into a soluble form you can work with, you need special agents. Agents that can disrupt those hydrophobic associations and essentially disassemble the lipid bilayer, but hopefully gently enough not to destroy the protein itself.
Okay, what kind of agents?
The most useful tools for this are detergents.
Detergents, like soap.
Sort of. Chemically, they're similar. They are small amphiphilic molecules, just like lipids, with a water-loving head and a water-hating tail. When you mix detergents with membranes at the right concentration, their hydrophobic ends bind to the hydrophobic regions of the membrane proteins, essentially coating them. This displaces the natural lipids surrounding the protein and brings the protein into solution as a protein-detergent complex. The detergent effectively shields the protein's hydrophobic parts from the water.
So the detergent acts like a temporary lipid substitute.
That's a perfect way to think about it. Now, there's a catch. Strong ionic detergents, like SDS, which you might know from gel electrophoresis, they will solubilize even the most stubborn hydrophobic proteins, but they also tend to completely unfold them, denature them, making them inactive.
Not very useful if you want to study function.
Exactly. But thankfully, there are also mild non-ionic detergents. These can solubilize membrane proteins while usually keeping them in their active, folded, functional state. And this is absolutely crucial for what are called reconstitution experiments.
Reconstitution, putting them back together.
Precisely. Imagine being able to take a purified membrane protein, kept happy in its mild detergent bubble, and then carefully reincorporate it into artificial phospholipid vesicle little membrane bubbles you make in the lab, or even into tiny, stable lipid patches called nanodiscs.
Wow, building a mini membrane system.
Yeah. These reconstituted systems are incredibly powerful. They allow researchers to analyze the precise activities of transporters or ion channels or signaling receptors, completely isolated from the overwhelming complexity of a living cell. For example, it was through reconstitution experiments like these that scientists could definitively prove that ATP syntheses, those amazing molecular machines, actually use the energy stored in a proton gradient to produce ATP. They rebuilt the system outside the cell to prove it.
That's incredible proving function by rebuilding it from parts.
Yeah.
Can you give another example? Maybe a specific membrane protein whose structure really opened up our understanding of how these things work.
Oh, certainly. Bacteriorhodopsin is the classic example here. It was the very first membrane transport protein whose full atomic structure was determined way back. And it became a landmark, really a prototype for understanding many other multipass membrane proteins discovered since.
Bacteriorhodopsin, what does it do?
It's found in the membrane of an ancient microorganism, an archaeon called Halobacterium salinarum. It lives in very salty environments and has these patches in its membrane that look purple.
Purple membrane. Cool.
Yeah. And that color comes from bacteriorhodopsin. It's a light-activated proton pump. Its structure showed 7 autohelices packed closely together spanning the membrane, and nestled inside is a molecule called retinal, which absorbs light. The structure perfectly revealed how a single photon of light hitting the retinal causes subtle changes in the protein's shape, which leads to the transfer of one proton, H+, from the inside of the cell to the outside.
Pumping protons using light energy.
Exactly. This light-driven proton pumping creates a proton gradient across the membrane, more protons outside than inside, and the cell then uses the energy stored in that gradient to synthesize ATP. It's effectively converting solar energy directly into cellular chemical energy.
Amazing. A tiny solar-powered pump.
It really is. And what's truly fascinating is that Bacteriorhodopsin turned out to belong to a huge superfamily of membrane proteins that all share a similar structure, those 7 transmembrane helices. But they have vastly different functions. This includes the rhodopsin in our own eyes, which detects light for vision but doesn't pump protons. It also includes the incredibly important G-protein-coupled receptors, or GPCRs. These are major drug targets. They sense hormones, neurotransmitters, odors, but they act as signal transducers, activating other proteins inside the cell, not as transporters.
Same structure, different jobs.
Right. Even things called channelrhodopsins, found in green algae, which are light-activated ion channels, not pumps, share in this basic structural theme. And scientists have now borrowed channelrhodopsins and used them as amazing tools in neurobiology to control nerve cell activity with light.
So that one structure unlocked a whole family of important proteins.
It really did, a foundational discovery.
Okay, so we have this fluid membrane, we have proteins embedded in it doing all sorts of jobs. You mentioned they diffuse laterally. Did these proteins just float around freely then, like little boats on this lipid ocean?
Well, we know membrane proteins like the lipids generally don't flip-flop across the bilayer. Those energy barriers are too high, but they do rotate around their axis and diffuse laterally within the plane of the membrane. The early, really elegant evidence for this came from experiments back in the 1970s, where scientists fused a mouse cell and a human cell together.
Fused cells, okay.
Yeah, they created a hybrid cell, a heterokaryon. Initially, the mouse membrane proteins were all on one half of the fused cell surface, and the human proteins were on the other half. But when they watched over time, within about half an hour or so at body temperature, the mouse and human proteins had completely intermixed over the entire surface of the hybrid cell.
They spread out. Proof of movement.
Exactly. Clear demonstration of lateral mobility. And this movement is absolutely crucial for many cellular processes, especially signaling, where proteins often need to come together to form active complexes and then separate again.
But that classic fluid mosaic model, the idea of proteins just floating freely in a liquid sea, that isn't the whole story anymore, is it?
Yeah,
Cells must have ways to organize things better than that, to create order out of this apparent chaos.
You're absolutely right. That simple picture is, well, significantly oversimplified. Most cells actively confine membrane proteins to specific regions. They create specialized membrane domains.
Domains, like neighborhoods.
Exactly like neighborhoods. A great example is in epithelial cells, like the cells lining your gut or your kidney tubules. These cells have distinct surfaces. There's the apical surface, which faces the lumen, the inside of the gut or tubule, and the basal and lateral surfaces, which face the underlying tissue and neighboring cells. And different membrane proteins, like transport proteins or enzymes, are strictly segregated. Some are only found on the apical surface, others only on the basolateral surfaces.
And that separation is important for their job.
Absolutely essential. For example, to absorb nutrients from the gut lumen, apical side, and then transport them out into the bloodstream, basal side, you need different transporters on each side. And this crucial asymmetry, this organization, is maintained by specialized intercellular junctions called tight junctions.
Tight junctions, what do they do?
They form a seal between adjacent epithelial cells, like a belt around each cell, and this seal acts as a physical barrier, literally like a fence, preventing both proteins and lipids from diffusing from the apical domain to the basolateral domain, and vice versa. They keep the neighborhoods separate.
Wow, cellular fences. That's clever. Are there other ways cells restrict protein movement besides junctions?
Oh yes, several ways. Proteins can sometimes self-assemble into large, relatively immobile aggregates. We saw that with bacteriodopsin forming those 2D crystals in its native membrane. Or proteins can be tethered. They can be anchored to structures outside the cell, like the extracellular matrix. Or, very commonly, they can be tethered to structures inside the cell. The most important one here is the cortical cytoskeleton.
The cytoskeleton just beneath the membrane.
Exactly. It's this dense meshwork of filamentous proteins, like actin filaments, located just underneath the plasma membrane. In red blood cells, for instance, the spectrin-based cytoskeleton is incredibly important. It forms this scaffold that attaches to membrane proteins. And this scaffold is crucial for maintaining the cell's unique biconcave shape and its remarkable mechanical strength and flexibility. It allows the red blood cell to squeeze through tiny capillaries without rupturing.
And if that cytoskeleton is faulty?
Then you get problems. Genetic abnormalities in spectrin, for example, lead to red blood cells that are fragile and spherical instead of flexible discs. This causes certain types of anemia because the cells break easily.
So this cortical cytoskeleton, it acts like a literal fence, or maybe more like a mesh, defining territories or corrals for membrane proteins.
Precisely. That's a great analogy. The cortical cytoskeletal network forms these mechanical barriers. It creates corrals. And these corrals significantly obstruct the free, long-range diffusion of proteins within the membrane. Researchers have used techniques like single particle tracking to watch individual membrane proteins moving. And what they see is that proteins diffuse rapidly within a corral, but they're largely confined there. They only occasionally manage to hop over the fence into a neighboring corral, maybe when the underlying cortical filaments transiently detach and reattach.
So restricts movement but doesn't totally immobilize them.
Exactly. It allows for local movement and interactions within a domain, but prevents large-scale mixing. And this corralling is thought to be really important for function. It helps concentrate signaling components together, for instance, making cellular responses faster and much more efficient than if everything was just randomly distributed.
Organization enhances efficiency. Makes perfect sense. Okay, one last major aspect. Membranes aren't always flat sheets. We know cells make vesicles, they have tubules, they change shape. How do they achieve this incredible diversity of shapes? How do they bend?
Right. Membrane shape is incredibly dynamic and actively controlled. And this control is fundamental for so many processes: vesicle budding for transport, cell movement, cell division. The list goes on. This precise shaping involves a whole cast of specialized membrane-bending proteins.
Membrane-bending proteins, okay.
Yep.
How do they work?
They attach to specific membrane regions and then deform the bilayer using one or more main mechanisms. One way is that some proteins insert hydrophobic domains, maybe an amphipathic helix, like little wedges into just one leaflet, one half of the bilayer. This effectively increases the surface area of that leaflet relative to the other one, causing the membrane to curve or bend away from the side with the inserted wedge.
Inserting wedges. Clever. What else?
Another major mechanism involves proteins forming rigid scaffolds. These proteins assemble on the membrane surface and either physically push or pull the membrane into a curve, or they stabilize a curve that's already formed. Think of the coat proteins like clathrin that assemble into cages to shape budding vesicles during endocytosis. They're essentially sculpting the membrane.
Right. The protein coat forces the shape.
Exactly. And a third mechanism involves influencing the lipids themselves. Some proteins can cause the local accumulation or depletion of specific lipids that have intrinsic shapes. Some lipids are more cone-shaped, others inverted cone-shaped. If you enrich one leaflet with cone-shaped lipids, wider head, narrower tail, it will tend to curve away from that leaflet. So proteins can bend membranes indirectly by changing the local lipid composition.
So they can bend it directly or indirectly via lipids.
Yes. And often it's not just one mechanism. Different membrane-bending proteins often collaborate, using multiple strategies together, to achieve a particular, often complex curvature needed for a specific cellular process. It demonstrates yet another layer of incredible complexity and coordination in how the cell organizes itself.
What a journey we've taken.
Yeah.
Seriously, from the simple yet, well, profound amphiphilic nature of lipids that just spontaneously self-assemble into this fundamental barrier, all the way to the incredible diversity and the dynamic organization of membrane proteins, the fences, the corrals, the bending. It's crystal clear that the cell membrane is far, far more than just a passive bag holding the cell contents. It's this bustling, highly regulated, and surprisingly fluid interface. It's essential for pretty much every aspect of life.
Indeed, we've really peeled back the layers today, I think, to see how membranes, even though they're just a few nanometers thick, unbelievably thin, are absolutely essential for everything. From generating cellular energy and receiving signals from the outside world, to interacting with neighbors, and even determining a cell's ultimate fate through things like apoptosis. The intricate interplay between the lipid composition, the protein structures, and those cytoskeletal interactions truly creates a remarkable and incredibly adaptable system.
It really makes you think, doesn't it? How does the cell orchestrate such precise control over all these constantly moving parts, the dynamic lipids, the proteins shifting around, interacting, the membrane changing shape? How does it coordinate all that to carry out specific functions? And what happens when that coordination breaks down? In disease, for example, it's like this continuous microscopic dance that underpins all of life as we know it. Well, thank you so much for guiding us through that. And thank you for joining us on this deep dive into membrane structure. We hope you've enjoyed this fascinating exploration into the microscopic world that quite literally makes life possible. Until next time, keep digging, keep learning, and thank you for being part of the Deep Dive family.